Welcome back to Open Med School. Today I am going to discuss with you about transport via vesicular mechanism, otherwise vesicular transport. As usual, I will give a small account of the vesicular transport and then go on with the MCQ. So take a cell. When it come across with a bacteria, this will produce pseudopodia, or otherwise called false legs. It will engulf and form a vesicle. So this will go like this and it is internalized. This process is called endocytosis. So here you can see there is an invagination occurring and later it forms a vesicle. So this method of engulfing large particles is called endocytosis. Endocytosis can be of three different varieties. First is phagocytosis. Phagocytosis, phago means eating. So a solid particle eaten by a cell, that is forming into a phagosome, that is phagocytosis. If it is a liquid, it is drinking and it is called a pinocytosis. Some cells are eating big particle and they are macrophages. Macro means large, phage means eating. Now we go into the third type of endocytosis, that is the receptor-mediated endocytosis. So here what is happening, certain materials like iron, LDL, they are internalized using this mechanism. So let us take the LDL. So this is a liver cell. Liver cell has got certain receptors called LDL receptors. When the cholesterol containing LDL reaches here, this will get attached to the receptor. And certain proteins called clathrins will help to make it internalized. So clathrin coated vesicles are formed. So here proteins called the clathrins. And the LDL with the receptor is internalized. From this, the LDL is used for metabolism, converted to VLDL and other materials, and the LDL receptor is sent back, it is externalized. So this is called the receptor-mediated endocytosis. Hope this point is clear to you. Now we think about the other type of vesicular transport, the exocytosis. Endo means inside, exo means outside. So This is usually for neurotransmission. So we'll take a nerve terminal. So here in the nerve terminal, the neurotransmitters are stored as vesicles. When the action potential comes here, calcium enters, and this will produce movement of these vesicles towards the synapse. So this is the synapse, and proteins like actin, film, and myosin will help for the movement. And this vesicle will fuse with the cell membrane, this membrane, and the neurotransmitters are released into the synaptic cleft. So transmission of neurotransmitters is an example of exocytosis. So today I have discussed about exocytosis and endocytosis. Endocytosis is divided into pinocytosis, that is drinking, phagocytosis, eating, and receptor-mediated endocytosis. Now we go to few MCQs. So the question is, a nerve cell releases neurotransmitters into the synaptic cleft. What type of transport mechanism? It is an active transport. It is not diffusion. The correct answer is exocytosis. It is not a simple diffusion. So the mechanism I have explained. So it is an enlarged view of the nerve terminal. So here you can see these are the synaptic vesicles which contain the neurotransmitters. It is when the impulse comes, it fuses with the cell membrane and contents are released. This is what I have said, the actin and myosin filaments help for the movement of the synaptic vesicles towards the cell membrane. Hope this is clear to you. And you can see the contents are released into the cleft. So neurotransmission is an example of exocytosis. Same way the glandular secretions, also this process is working. Again, this is showing the actin filaments and the myosin, which is helping for the synaptic vesicles to fuse with the or move to the cell surface. And this is the release of the neurotransmitters. The question number 2, in a macrophage, which transfer process is responsible for engulfing and digesting cellular debris? The correct answer options is, it is not autophagy, it is phagocytosis, it is not exocytosis, it is an endocytosis, but the best answer option will be phagocytosis. Hope this point is clear to you. So again you can see here, this is the, suppose a bacteria comes, there is a pseudopodia develops, it engulfs, it goes to the vesicle, and later it fuses with the lysosomes, enzymes, and is being destroyed. And the residual things can be released by exocytosis. So hope you understood. Phagocytosis, eating, and pinocytosis, drinking. Pino is drinking. So endocytosis, as we have said, it can be phagocytosis, pinocytosis, or receptor-mediated. So this is what I said, there are certain receptors like LDL receptor or receptor for iron. Once it is this attaches with the receptor, the LDL attaches with the receptor and it gets internalized, and then the receptors are sent back to the cell surface. and there are certain proteins called clathrins, and this receptor-mediated endocytosis is helped by clathrin protein. So let us think about a clinical situation. The classical example is familial hypercholesterolemia. It's a condition where the LDL receptor is abnormal. That is, in the cell I have told the hepatocyte, there are receptors called LDL receptors. The LDL receptor, there is a mutation is occurring. So the LDL is unable to attach with the membrane. So what happens? The serum level of cholesterol increases. And such cases, if left untreated, 50% risk for fatal, non-fatal coronary events by the age of 50 years. So they can develop myocardial infarction, because the myocardial infarction is due to deposit of cholesterol in the blood vessels and blocking the blood vessel. So familial hypercholesterolemia is due to a defect in the LDL receptor-mediated endocytosis. That is the clinical relevance. So this is the pinocytosis about which we have discussed, that is drinking. Along with the fluid, some amount of material which are soluble in the fluid can also be ingested. So we have discussed vesicular transport, which are divided into endocytosis, exocytosis. Endocytosis divided into pinocytosis, phagocytosis, and receptor-mediated entry, and hypercholesterolemia. The familial hypercholesterolemia is an example of a abnormality in the vesicular transport, receptor-mediated endocytosis. Hope it was useful to you. Stay tuned. Thank you.